My name is Tugabirwe Joan. Today I'm going to take you through banana propagation, and here at Rwentanga Farm Institute we propagate our banana tissues under a humid chamber. This is our humid chamber. In our humid chamber we are able to produce the clean varieties, selected and disease-free varieties. So I selected my suitable site for the humid chamber. I cleared the vegetation. After clearing the vegetation, I measured the area of 1 meter width and a reasonable length. After measuring my area, I made the holes for the poles. After putting the poles, then I supported the sides of the humid chamber with the pieces of timber. After that, I laid a black polythene paper on the bottom of the chamber on the leveled ground. Then I put a layer of sawdust on top of the black polythene, and the purpose of the black polythene is to absorb sunlight and enhance root development. After putting a layer of sawdust, I obtained my planting materials and prepared them. I planted them in the sawdust. After planting my materials in the sawdust, I covered them with another layer of the sawdust on top. After covering them, I got my white polythene paper and covered the whole chamber to make sure it is airtight. So I'll be watering my chamber for a period of 1 month. After 1 month, I'll be expecting the sprouting of the plantlets out. So after the sprouting, I'll get my plantlets and put them in the bags. Now we are at the potting stage from the humid chamber. The plantlets are uprooted from the humid chamber and put in the potting bags. So this tissue was obtained from the plant chamber, the humid chamber. I uprooted it from there and put into the potting bag. So the process of putting this tissue in the potting bag, you first obtain the soil types, different soil types. You obtain the topsoil, the subsoil and manure. You mix the soil types in a ratio of 2 to 2 to 1, meaning that 2 parts of topsoil, 2 parts of manure and 1 part of sand soil. You fill the mixture in this potting bag, and a plant tissue is gently uprooted from the mother comb into the potting bag. You water immediately. Make sure the watering takes place immediately. Then you transfer the potted tissues into the nursery bed, from where they are cared for 1 month before being taken to the main field. From the humid chamber, we potted our plantlets and put them in the nursery bed. So from the nursery bed, we planted them in the seed bed, and this is the final stage of planting in the seed bed. So we prepared our seed bed early in advance. In our main garden, we prepared it in the following steps. Firstly, we sited the suitable site, which is this one, and we did the pegging. The pegging was done by measuring a distance of 3 meters by 3 meters, meaning that it's 3 meters wide and 3 meters long from one plant to another. After pegging, we dug the holes, and the hole was measuring 3 feet by 3 feet, meaning 3 feet wide and 3 feet deep. After digging the holes, we mixed the soil with manure and filled the holes. After filling the holes, we brought our planters from the nursery bed and planted in those filled holes. After planting, we did the watering, and to continue with the watering, we do the weeding, we do the pruning, and other practices so that we manage our garden very well as we are waiting for the first harvest after a period of 9 to 12 months.